In this video, I'm going to demonstrate how to experiment osmosis using a potato. So this potato, though yes, I use it domestically. I use it to make porridge and all. It tastes very nice. So it contains living cells, and these cells, this potato, for example, potato yam, and some nice carbohydrate can be used to demonstrate osmosis. Okay, so the aim is to demonstrate osmosis using a potato. All right. So the materials needed. So you need the potato, slice the potato, you need the salt. So this salt serves as the strong solution and strong solute. So you put in the water, become a strong solution. Then you need the water, which is a universal solvent, and in two containers, you can use one for a strong solution and the other one for a light solution or a soft solution like that of normal distilled water. Then your knife. So you use the knife to slice the potato. Okay. So So the procedure that you need to go in this is the first thing you need to do is to cut your potato into pieces. So the both potato have to be in the same size so you can get the initial result. So the size of each potato have to be the same. Okay, so you can get the desired result you need. So the next thing you need to do, put one potato piece in water. So you can put one in normal distilled water. So this water is not a strong solution, just in normal distilled water or your tap water in that case. Then the next step, you put the other potato piece in the salt water. So you add salt to the water, it becomes a strong solution, become a hypertonic solution. So you add salt to it, and you add your piece of sliced potato into the solution. Then the next one, all you need to do is to leave both specimen for 30 to 60 minutes, and you're going to see what's going to happen. So you're going to check and observe what's going to happen. So for the step 5, remove both of them and compare and see what happened to the shape and the size of both of them to see if they remain the same or if one becomes smaller or the other one become bigger. So what to confirm? Well, let me just quit talking too much. Let's go to my kitchen where I'm going to demonstrate how this experiment is done. Okay, so follow me to my kitchen. Let me show you how it's done. Okay, so this is just a simple setup for the osmosis experiment. So I'll be using this is my kitchen. So this is not really in a real laboratory settings, but it's just kind of like illustration, something you can do at home and confirm and see if it's going to happen. So now I'm going to put a water here. I'm just going to fetch it from the tap. Put the water to this point. Okay. So this one. Then I'm going to put water here also. So I'll try to make sure they're on the same level. So one other thing we need to do is so this water that doesn't contain salt. So this one is hypotonic solution. While this one contains salt, so I'm going to put salt solution here. I'm going to put a salt solute. So this one contains high amount of salt. So this is a hypertonic solution. Okay. So all I have to do, I just let it dissolve or try to stir it. So I'll just stir it so that the sauce can dissolve properly. Good. Then the next thing I'm going to do right now, this is just my sliced potato. This is my sliced potato. So I'll put each slice in each of them. So this is normal water, while this is salt water. Normal solution, while this one is a strong solution. So I'm going to put one inside the normal water. I'll put the other into the salt solution. I'm going to observe this for like 30 to 1 hour. I'm going to leave for 1 hour. Then I'll come and check and see. So one thing you need to confirm is the size of the potato have to be the same so you can easily see the difference between both of them. So I'm going to come back and check it after 60 minutes and see if there be any differences between both of them. So after 60 minutes, so let's check and see what happens between both of them. So don't forget that this one contains normal water while this contains strong salt solution. So if I bring the first one out using my spoon, so if you look at this, you can see that even the potato have actually increased in size. Why? Because
it has absorbed more water and become more turgid. So it looks more bigger, looks bigger in size. Put it this way, it looks bigger in size while you have absorbed more and large amount of water, and the cells is so turgid. So if you look at it, the solution is hypotonic because it's just normal water. It's not as strong as that of the saltwater solution. So if I bring out the other one, then you can see clear big difference. You can see that the potato have reduced in size. It has reduced in size. And if you look at the surface here, you can see that you can look at it that it even looks somehow peeled. Why? Because the potato have lost a lot of water. Because why? The solution is hypertonic, and in that hypertonic solution, water tends to what leave the cells, and once it leave the cell become what plasmolized. So if you look at it, this look at the size, it become what smaller. If I bring that of the normal water to both of them, you can see. If you look at the size, both of them, you can see this one is bigger. Why? Because I've absorbed more water. If you look at this one is so smaller because water have actually left the cell and it has become what? Flaccid. So this one is more turgid and bigger, while this one is more flaccid and smaller. Okay, so this undergoes what we call um, uh, it's not really hemolysis because it's not really red blood cells, but that receive a large amount of water have become turgid. Why this one have lost more water and this one become flaccid. So this potato will be plasmonized because what large amount of water will be reduced because it was placed in a hypertonic solution. Okay, so now let's go back to the classroom.
So you can see the differences that occur between both specimen. All right. So just to give an overview of what I've just explained. Okay. So the potato in water, you said the potato become hard and firm. Why? Because it has absorbed a lot of water and become turgid. So it become hard, very hard and firm when you touch it. Very hard and firm. So why? Because water move into the potato cells by osmosis. So that's the reason. And again, the potato in the salt solution become very soft and flabby. That's why if you look at it, it has actually reduced size. Why? Because it lose a lot of water from the cell. So water moves out of the potato cells by osmosis. Very good. So just to give you an overview, if you look at this picture here, so this is A and B. So this the first one is concentrated salt sugar solution. This is A1 and A2 concentrated sugar solution. So if you look at it, this is the initial and this is the final. Okay. So if you look at this experiment here, it does give you an overview of what's happened. So if you look at this B1 here, at first the cell the cell is actually large in size, okay, but in B2 when water molecule have actually escaped from the cells, what happened? It become flaccid that actually was reduce size drastically. Okay, so that is what this thing is all about. So it just give you the let you know that okay, once you place the cell in a strong solution, what happens? The water tend to what escape from the cells, and escape from the cells, then the initial mass of that cells tend to to reduce. So some questions that we can ponder about. So what process was demonstrated? Meaning the experiment, what process? So this is osmosis. Just demonstrate osmosis. What happens to the potato in the water? In the normal water, it becomes firm. And why does it become firm? Because it has actually absorbed water. So water enters the cells and the cell become turgid. So it becomes very firm. And what happens to the potato in the salt solution? So it becomes very soft. It becomes soft and flabby. Okay. So why does it become soft? Because water leaves the cells. Once it leaves the cells become soft. And which solution contains more salt? And don't forget it, that's a salt solution, which is also known as what? A hypertonic solution. Okay. So that's that for the experiment for osmosis. It's a straightforward experiment. Okay. So I'm going to see my next video where I'm going to solve some real life experiments on real life questions on osmosis and diffusion on how you're going to see them in an examination. Okay, so my next video, just keep an eye on my channel where I'll upload a video that explains multiple choice questions and some theory questions on osmosis and diffusion.